And we're starting, so what does that all have to do with drug discovery? Well, there are a series of initiatives. How many people here have heard of Sage Bionetworks? Okay, so this kind of brings together a number of the things that I've been talking about. You know, the use of the commons, which puts into the public domain lots of biological data, genomic data, patient data, and everything in between. There is an open framework, there are open licensing to make all of that accessible. And so this is disease models that are going online and being accessible. And so the idea, it's founded by Stephen Friend, who actually founded Rosetta Pharmaceuticals, which was a spin-off from Merck. So, you know, it's someone who's been along the traditional route and now is dedicated to the idea of creating open drug discovery capabilities. And I think that's kind of an example of bringing that together. When I gave this lecture last year, we were actually just in the point when, if you remember, Wikipedia was closed down for a while, and there was a huge ruckus, and I think that's just another sign that the open access battle was being fought, and it's not been won yet, but it's kind of looking good from my point of view. All right, so what's all this? We've sort of seen what's going on. We've seen the notion of a Sage Bionetworks as an open consortium for drug discovery. How could this potentially impact drug discovery more directly? So I don't want to necessarily answer that question, but I want to go back and I want to look at the question is why is the drug discovery process, why are we not discovering many new drugs? There are economic reasons. I want you to tell me, but you can't tell me. But you could tell me, but you can't tell me because of, for whatever reason. But here's a couple of data points that got to get you to start thinking, all right? Gene knockouts. When you knock out a gene, only in about 10% of cases do you actually see an effect on the phenotype. Why is that? Because there's redundant functions. There's alternative network routes. There's robustness of the interaction networks. So that's already got to ask you, so okay, you're knocking out a gene. What if you, in some way, perturb the system with a drug so you hit a particular target? Well, in many cases, because for these exact same reasons, you might be having very little effect. You know, you can't ignore these things, all right? And then when you look at the fact that if you take all of the biologically active compounds and you start looking at them and you assay them in various ways across panels, you find just from that group, 35% of them bind to more than one target. So this whole idea of the premise of the drug discovery paradigm for so long with respect to small molecules of one drug, one target, one disease, this kind of data just says, how can that possibly work? To me, it explains exactly why when I turn on the TV and try and watch the news, most of the time I'm hearing about the side effects of drugs that are being advertised during the commercials. And the reason there are these side effects, in large part, has to do with these kinds of effects. All right, so what should we do about it? What has the drug industry done, in my opinion, for quite a long time? It tried to ignore it. It kept trying to chase after these blockbusters that were these, and there are a few out there. Many of them being discovered serendipitously after all of this massive amounts of research, but they're all out there. And a lot of them are actually natural products. They're not actually synthesized drugs. So, but if we accept this notion that there isn't one drug, one target, one receptor, what can we do? So here's something that actually drives this home even further, all right? This is the human genome tree. So you and I have about 550 human protein, we're humans, so we have 550 protein kinases, approximately. This is the phylogenetic tree of those kinases. Each one of these instances is, and the balls on it are kinase inhibiting drugs. They're on the market, FDA approved drugs. And so you can see, Gleevec, which is a wonder drug, and the size of the ball here is a reflection of the binding affinity to a target. A number of these drugs actually bind to off targets with higher affinity than what the known target was believed to be. And then you take something like staurosporine, which is a natural product, that binds to huge parts of the tree. So what does that mean? It means that you're not inhibiting a single target. What you're seeing is a massive collective effect. So then the question becomes, well, how can we use that? If we accept that notion, what can we do with it? So this Ehrlich philosophy of the magic bullet is, I think, is not being realized. So it's just naive in a complex system, in my opinion. All right, so how can we apply that? How can we accept that? And then what can we do about it? So I'm going to give you a couple of examples from our own work. All right? So if we accept the fact that we've got this multiple binding, let's try and figure it out, what it is. Let's say I've got a drug, it's this right fist, I've got a receptor, and these things are binding together. I know what that receptor looks like now. I know what its binding pocket looks like because I have the complex. So let me take that binding site and let me search across the complete structural proteome, the proteome of the human, looking for all instances of a similar binding site. If I find that, then it tells me one of two things, three things. It may tell me nothing, but it might help explain the side effect of a drug because I now know another target. But even better, from a drug discovery point of view, it might actually allow me to repurpose that drug because I've discovered a target that's actually associated with a different condition. And I have examples of all of those. I'm going to try and tell you a little bit about a couple of them. So these are the kinds of things it tells you, and I pretty much described that. So the basic idea is in this database I mentioned, there are things like Lipitor, $100 billion a year drug. It's sitting in a public database. And so you can go, in this PDB I mentioned before, so you can go and look at it bound to its target, and you can play around with that. So you can take that target, and you can look for other similar targets. And so, there it is. And so what you do is you characterize the binding sites, and then you essentially search for those binding sites across all these other proteins, and you do the statistical test to see how valid what you discover is. How valid is this alternative binding site statistically? I'm not going to go into any of the science of this. This is work that's been done by Leger in a lab. I just want to say that. And, so that's sort of the technology. I'm not going to go into any of the details. So what can you do with it? What are the kinds of things you can do with that technology? So we happen to be interested in neglected tropical diseases, partly because, of course, the reason they're neglected is hardly anyone is really looking at them. The drug companies are not, until recently, haven't been looking at them very much because there's no money in it. And yet, on the other hand, there's 1.7 million deaths a year approximately from tuberculosis. We also, people worry about it constantly because we have multiple drug resistant and we have extreme drug resistant strains that, could eventually get into this is a developing world disease, and unfortunately it doesn't get the attention it should. But if it gets into the developed world and these strains start really proliferating, we've got a lot of problems because there hasn't been, until recently, particularly successful new treatments for TB for 40 years. So this just happens to be something that we would focus on.